200 million gametes enter an acidic chemical war zone, and 99% of them perish almost immediately. While we've long understood the basic biological premise of reproduction, the exact mechanics of how these survivors navigated this dark space remained a mystery for decades. Early researchers were physically constrained by their tools. They relied on two-dimensional microscopes that offered static, low-resolution glimpses of a highly dynamic environment. Solving for these mechanics required researchers to look past the lens of a single microscope and instead analyze the movement as a series of interconnected data points. The journey begins with a microscopic GPS. The egg releases specific chemical attractants into the fluid, laying down a trail for the survivors to follow through the darkness. This visualization shows how researchers locate those specific signals. It plots 40 million biomedical citations from PubMed, filtering down into a concentrated cluster of specific chemical research. That literature points to databases like PubChem, isolating the exact molecular structure of the egg's chemical attractant. Without this data-verified chemical roadmap, the remaining sperm would swim blindly into the void and fail to reach their target. Nearing the egg, the sperm undergoes hyperactivation. The tail's smooth swimming wave suddenly shifts into an aggressive, asymmetrical whip to generate forward thrust. Observing this multi-dimensional physics in true, verifiable 3D architecture was finally made possible by tools like the Journal of Cell Biology's Data Viewer application. This three-dimensional model breaks down the sperm's movement using Z-stack image data. Rendered in strict RGB color standards to preserve biological accuracy, it separates the motion into distinct optical layers. Viewing the structural geometry from every angle proved exactly how the sperm generates enough physical force to breach the egg's thick outer defenses. The sperm strikes the zona pellucida, the egg's protective outer barrier. This collision triggers the acrosome reaction, violently rupturing the sperm's outer membrane. Stripping away that membrane exposes the ultimate biological challenge, identifying the precise, unknown proteins responsible for fusing the two cells together. To find them, researchers sequence the genome and submit those raw, untranslated genetic reads into the GenBank repository. NCBI BLAST cross-references the unknown genetic string against a massive database until hitting a match. The vast database translates this linear genetics into a complex 3D biomolecular protein, revealing the specific docking between Izumo1 on the sperm and Juno on the egg. This identification of the exact biochemical interaction wasn't an isolated observation, but the specific breakthrough needed to explain the logic of fertilization. The moment Izumo1 and Juno dock, it triggers a massive, instantaneous wave of intracellular calcium across the entire egg. In immediate response, the egg sheds all of its remaining Juno receptors, dissolving them into the surrounding fluid. This initiates the cortical blockade. The egg's outer shell hardens permanently to block any other sperm from entering, securing one unique genetic blueprint and preventing lethal polyspermy. This precise and ruthless biological cutoff guarantees the safe survival and development of exactly one new life. Once the biological sequence of fertilization is complete, the process of documenting the discovery for the public record begins. Submitting research to journals like Science or Nature means facing a 94% desk rejection rate, which filters everything except findings that significantly change a field's perspective. The final, validated results are archived at the National Institutes of Health, creating a permanent home for the discovery within NCBI's bookshelf and PubRed Central. Just as the egg's hardening process secures a new genetic blueprint, the validation of this research secures its place in history, turning a competitive scientific effort into a permanent addition to human knowledge.